Non-animal human-based technologies have undergone a massive development over the past decade. Nonetheless, around 5 million animals are still suffering in German labs every year, despite the lack of transferability of animal data to humans. Other countries all over the world have already developed strategies to phase out animal testing, amongst them the Netherlands and the USA. We also observed developments in the European Union. The European Parliament passed a resolution calling on the European Commission to develop a concrete phase-out plan for animal testing. Also, more than 1.2 European citizens advocated for a Europe without animal testing via a European Citizens Initiative. Unfortunately, the German government has not shown major efforts to move in that direction so far. In order to change that, Doctors Against Animal Experiments and People for Animal Rights Germany are running the campaign Phase Out Animal Experiments Now, calling for a concrete strategy to phase out animal experiments in Germany. As part of this campaign, we organized today's event with 3 experts explaining the situations in the USA, the Netherlands, and the European Union, and demonstrating the possibilities of phasing out animal experiments. I am proud now to introduce our 3 brilliant speakers. Dr. Aisha Akhtar from the Center of Contemporary Science in the USA, Professor Merel Ritskes-Heutinga from the University of Utrecht in the Netherlands, and Tilly Metz, member of the European Parliament from the Greens. A big thanks to all 3 speakers for joining us today. We will have a Q&A session at the end of the event, so after the 3 presentations. Please place your questions in the chat. We apologize if we might not be able to answer all questions within the limited time frame. So I will now introduce our first speaker, Dr. Aisha Akhtar. Aisha is double board certified neurologist and a specialist in preventive medicine. She is founding member and chairwoman of the Center for Contemporary Science in the USA. Aisha, thank you so much for being here, and the floor is yours.
Thank you so much, Tamara. So let me try to share my screen, and I'm going to make sure. Can you confirm if my screen is showing presenter mode?
Yes, everything is good.
Okay, good. Thank you. So thank you everyone so much for having me here. I just give you a little bit of background of myself. As Tamara mentioned, I'm a neurologist. I've actually been involved in the work of trying to replace animal testing for more than 30 years, since 1988. I was in high school at the time, and I've been involved in this field since then. And I entered the field to replace animal testing initially because of the animal ethics issue. The suffering of animals is a tremendous problem in this world, and I initially got involved because I care so deeply about ending the suffering of animals at human hands as much as possible. And despite the fact that I had worked on trying to, with many others, trying to showcase the suffering of animals and trying to talk about the ethical issues surrounding the use of animals for experimentation. In the past 30 years, for the most part, with some exceptions, that approach hasn't made too much of a difference in the numbers of animals used in experimentation. And let me just make sure I can move my slides. Okay, so if we look at the number of animals used worldwide, indications are that it's actually growing. More animals are used today than were used 10 years ago in experimentation. The best thing we can do is estimate how many animals are used, and that is because in many countries, and unfortunately the United States is one, there is very little transparency, not only on how many animals are used, but how they are used, which animals are used, what kinds of experimentation are they used in, and how our tax dollars is used to support the animal experimentation. But the latest estimates that I found is that at least 200 million animals are used every year worldwide. At least 100 million are used in the United States every year. And the top 3 countries that conduct experiments on animals are the US, Japan, and China. So because of the fact that the numbers have been growing, despite attempts to try to highlight the suffering of animals, I really started to shift my approach to some degree. And the approach has also been informed by my experience, not only personally, but also my experience as a physician. I can't tell you, as a neurologist, how difficult it is to tell patients after I have diagnosed them with Parkinson's disease or told a family that their parent has Alzheimer's disease, but then to be able to say that I'm sorry, we have no treatment that can really affect the fundamental disease. We have treatments that may help with some of the symptoms of the disease, but nothing that really treats the disease itself. And then when I was at the Food and Drug Administration, I was there for 10 years. And during that time, I saw again and again how drugs and vaccines came through the pipeline again and again. Everyone would be excited that this is going to be the next hope for stroke or the next hope for HIV, only for them to fail when tried in clinical trials. After I worked 10 years at the Food and Drug Administration, I then moved to work as the Deputy Director of the U.S. Army's Traumatic Brain Injury Program. And despite the fact that head injury, traumatic brain injury, is a major problem, not just for U.S. soldiers, but soldiers worldwide and non-soldiers, of course. And despite the fact that our military has spent arguably hundreds of millions of dollars every year conducting head injury experiments in animals in the lab, there is not a single effective treatment for head injury. So that experience, in addition to the experience I've had with my own father, has caused me to rethink things differently. So my father, my Abu, as we call him in Pakistan, has experienced lifelong diabetes. And as a result of his diabetes, he frequently experiences neuropathy. And if any of you have ever experienced neuropathy, you know how excruciating this pain is. It's an inflammation of the nerves. And it's not a typical pain. It's very, very different, very uncomfortable, hard to treat pain. And I have seen my father again and again suffer from neuropathy and not be able to offer him any treatment that really handles his pain. So both my professional and my personal experience with my father has caused me to reframe the ethics, to not only ask, is animal experimentation ethical for animals, which of course it certainly is not, but is it even ethical for humans? And to ask that, and to answer that question, we have to ask, is it an effective use of resources, and does it reliably inform human health? If we look at how the drug and vaccine development process occurs in the US, we have to look at the Food and Drug Administration, the FDA. The Food and Drug Administration, of course, is the organization, the governmental organization that will oversee and approve medical devices, drugs, vaccines, tobacco products, and many nutritional supplements and ingredients for the market, for sale in the market. Now, the Food and Drug Administration requires, with rare exception, actually, I don't know if there's any exception, for all new, what they call investigational new drugs and vaccines, it requires that every drug and vaccine must be tested on 2 different species before it can consider being moved to clinical trials. Usually, one of the species is a rodent, a mouse or a rat, for example, and the second species is a larger mammal such as dogs, pigs, or monkeys. I don't know where this two species idea came from. And to me, when I look at this, it really doesn't make sense, because what makes us think that two species is going to be any more effective at predicting human outcomes? And what makes us think that using mice and rats as the one species, or using dogs, pigs, or monkeys as the second species, is going to improve the predictiveness of this phase of testing? You know, we are not mice. We are not overgrown mice. We are not overgrown monkeys. Despite the fact that my husband likes to tell me that I often act like a monkey, I am not a monkey. I am not a mouse. I am not as cute as this little monkey here. I am not even another human being. And we know that. We know that we do not necessarily predict what's going to be safe and effective in another. And it is for this reason why we must have diversity in human clinical trials, because what may be safe and effective in me may not be safe and effective in a 60-year-old Caucasian male, or safe and effective in a 12-year-old boy from Sudan. So we know that there's so much diversity, even within the human population, in regards to how well we respond to medications and other treatments. And we now know that despite the fact that the FDA has this requirement, this basic requirement for all new drugs and vaccines to be tested on 2 different species, there is a major problem. Because despite that requirement, up to 90 to 95% of all vaccines and drugs that pass the animal tests end up failing in human clinical trials. And there's a couple of things I want to point out here. So, you know, in the preclinical phase, before human clinical trial phase, we have not only animal testing, but we also have computer modelings and cell cultures and other types of testing methods. So you could say that the preclinical general is non-predictive of human results. The problem with that is it's actually one type of testing method that is predominantly the method that is used to decide whether or not to move a drug or vaccine into human clinical trials. And this I tell you from experience in institutional review boards, my experience in institutional review boards in which we reviewed and either approved for clinical trials to occur, both at academic centers and at the FDA, and in my 10 years of experience at the FDA. It is not the computer modeling, it is not the cell culture results that truly form the basis for the decision as to whether or not to allow a drug or vaccine to continue to move on down the pipeline and into human clinical trials. It is the animal testing results that form the basis for this decision. And we now know that about 30% of all drugs and vaccines fail in humans because of toxicity, and about 70% because they are ineffective. So putting these 2 points together, that it is mostly the animal testing results that form the basis for whether or not a drug or a vaccine will move into human trials, and looking at that 90 to 95% failure rate, it is very accurate to say that 90 to 95% of all the animal testing for drugs and vaccines does not predict, or that they fail in predicting whether a drug or vaccine is going to be safe or effective in humans. Now you think about that, a 95% failure rate. Could you imagine if you were to jump on a plane and the pilot said, all right, ladies and gentlemen, strap in because we have less than 5% chance of landing safely at our destination? Everyone would immediately jump off and demand a complete overhaul of the entire airline industry. But for some reason, we accept this failure rate when it comes to the very drugs and vaccines that we put into our own bodies. If I were to just also give a few examples of the immense failure rate in animal testing, thus far, there's been more than 700 treatments have been found effective in animal experiments in stroke. Of those, more than 150 have moved on to human clinical trials, and not one of those treatments has proven effective in humans. For stroke, we still use a treatment that we've had for decades, and it's marginally beneficial and only within a very narrow time frame. So we really, despite all the animal stroke experiments, we have not made any major progress in treating strokes in humans. If we were to look at HIV vaccines, for example, there have been more than 90 vaccines that have been found effective in animal experiments. And most of these experiments include non-human primates, and for many decades included chimpanzees. Out of those, more than 30 have been tried in humans, and not one has proven effective in humans. In fact, 2 of the vaccines were suspected of actually increasing the risk of HIV in humans. And in 2020, after more than 10 years was spent in just organizing the clinical trials for one HIV vaccine, and after the US government spent more than $100 million alone just on this one vaccine, and after everyone thought this was a vaccine that was going to work because it worked in non-human primates and in other animals, it again failed when tried in humans. So we can go down the line again and again. If we look at diseases, we can see that more than 172 treatments for Alzheimer's disease that were found effective in the animal experiments failed when tried in humans. More than 150 treatments for inflammatory diseases that were successful in animals failed when tried in humans. So there is a need to move away from animal testing, not only for the animals' benefit, but also for human health benefits. Here's a slide that kind of captures the drug development pipeline here in the United States, and I suspect this is probably pretty indicative of the pipeline throughout the world. But if you look at the different phases of testing, about 10,000 compounds may be screened in the drug discovery phase, and where at this phase, they are screened just to see if they could potentially be useful for a certain particular target, a certain disease or a certain aspect of a disease. Of those 10,000 compounds, approximately 250 on average will move into the preclinical phase. This is the before human clinical trial phase. Of those, maybe about 5 will move into clinical trials, and then of course, this slide says that 1 in 5 actually get approved by the FDA. It's actually 1 in 10 actually get approved by the FDA. So this isn't fully accurate. It's really less than 10% of drugs and vaccines actually get approved and into the market. But what I want to point out is the phase between the preclinical and the clinical trial phase. We now know that animal testing is non-predictive of whether a drug or vaccine is going to be safe and effective in humans. And so we see the result of where that failure happens towards the end of the drug development pipeline. But what I want to point out is the problems that may occur earlier in the drug development pipeline. If you think about this between the preclinical and the clinical, so about 5 compounds out of, let's say, these 250 compounds that are tested at the preclinical phase may move on to be tried in humans. And the question I have is, what happened to those 245 compounds, on average, that did not get tested in humans? How many of those actually would have worked in humans? How many of those would have been safe in humans, but were never even given the chance to be tried in humans because they failed the animal testing phase? And so this is an incredibly important question we have to be asking ourselves. How many missed opportunities, how many treatments, effective treatments, maybe even cures, were abandoned because of misleading results in animals? So of course, we'll never really know how often this may have occurred, but we have some data to suspect that this is not a rare event, to suspect that this may actually be routine. And I'll point to 2 drugs. So, or actually one drug in particular is cyclosporine. Cyclosporine is a drug widely used to treat a host of autoimmune diseases and organ transplant rejection. It's effective in these two areas. It's quite effective, and as I said, it's widely used. But when it was going through the animal testing phase, there were results that were very species specific that almost derailed the further development of cyclosporine, in particular with dogs. There were results in dogs that almost caused the abandonment of cyclosporine. Fortunately, it was caught that the result that was found in dogs was only specific to dogs, and the cyclosporine was allowed to continue on through the pipeline. And so we have it now on the market, thankfully. But there's been other drugs like cyclosporine that were almost abandoned because of species-specific results that did not apply to humans. And this has occurred a number of times to make us truly question how often has this occurred, and how many really useful drugs and vaccines have been abandoned that should not have been abandoned if we had better testing methods. So if I were to capture the overall harms to humans as a result of using a system that is based on animal testing results, we can capture three basic harms. One is that there are adverse events, safety problems that are not predicted by the animal tests. We know about 30%, at least in the US, about 30% of all drugs and vaccines do not get approved for the market because they are too toxic. They end up having too many adverse events or serious adverse events in clinical trials that were not predicted by the animal testing. The second main human harm is a tremendous wastage of time, money, and resources. Bright scientific minds out there, if instead of wasting time going down the same avenue again and again, doing head injury experiments on animals, trying to do HIV vaccine experiments on animals, trying to do stroke experiments on animals, if instead of going down that same failed line of investigation again and again, they use better methods and they use their time in a more fruitful avenue, think how much further ahead we could be in medical science. And think of all the wastage of money and other resources that go into these failed lines of investigation. I mentioned that one HIV drug, the vaccine that failed in 2020, $100 million alone was spent just on this one vaccine alone, and more than 10 years was spent just on the clinical trial phase of this vaccine. That's a lot of wastage of time and money and resources. And then, of course, the third harm to humans is the likelihood of missed opportunities, the abandonment of drugs and vaccines and other therapeutics that would have been safe in humans, would have been effective, but they were discarded because of misleading results in animals. So the solutions. So this is where we're going to get to the policy work. So the solutions we have that I would offer you, that we work on, and at the Center for Contemporary Sciences, my organization, is we work on educating the next generation of biomedical scientists. We conducted a pilot study of students, of biomedical students. And we found that all, except for one, and I don't know who this one was, every single one of the students wanted to learn about human relevant testing, would rather train in other human relevant methods rather than be trained in doing animal experimentation. But more than half of them had said that they didn't know of any opportunity at their university, or there was no opportunity for this kind of training at their university. Now, I suspect that number is actually higher because this pilot study was conducted with students from a well-equipped university that did actually have some programs to train students in like organ chip technology. So most universities currently, I suspect, do not offer training in these alternatives or human relevant methodologies like organ chips, artificial intelligence, and other types of testing methods. And so that, in that sense, the students are not being prepared for where the research is actually going, where the science is going. The science is going to move towards more human relevant testing methods. And so by not training biomedical students in these fields, we are not preparing them for the future. The second thing we do as an organization at the Center for Contemporary Sciences, we help to increase investment and funding into the companies that are creating these technologies. So we work with investors. We last year held the first ever investment conference in this space to help match investment with companies that are creating more human relevant testing methods. The third big thing we do is to change the narrative. And this goes back to the second slide I had about reframing the ethics about animal experimentation. We have to change the narrative. The narrative right now that has been put forth by those who have a vested interest in keeping animal experimentation alive, no pun in words there, but they have put forth this narrative that the public largely believes, that we have to experiment on animals for our own health, that our children's lives, our family lives depend on continuing experimentation on animals, when the evidence is really suggesting the opposite. That no, in order to best protect our health and the safety of our family, we have to move away from animal testing. The biology of a rat, of a dog, of a non-human primate does not reproduce the biology, does not model the biology of a human. And so we need methods based on human biology. So at the Center for Contemporary Sciences, we work to change that narrative, to show that moving away from animal testing not only is beneficial to animals, but it's beneficial for human health. It is where the future of medical science is going. So it is inevitable. Now the question is just how fast do we get there? And then, of course, the fourth way that we work at the Center for Contemporary Science is to help update policies and regulations. So let me go into the most recent policy. We call it a success, but I'm going to give some caveats. So just in December of 2022, President Biden signed into law the FDA, the Food and Drug Modernization Act of 2.0. And this law occurred in pretty much record time. The bill was introduced in just a little bit, it was introduced just about a little bit over a year prior to this actually being signed into law. And that's pretty unheard of, especially in the political climate here in the United States. Now, I helped draft some of the language of this bill, now law, and met with and helped recruit the two primary co-sponsors on the Senate side, Senator Cory Booker and Paul Rand. And at Center for Contemporary Science, we help provide some of the supporting facts, statistics, and arguments for this bill. And basically what this act does is it goes back to a 1938 law. In 1938, a law called the Federal Food, Drugs, and Cosmetic Act was enacted. And this was in response to several different events that had occurred. And one was an event, a tragedy, in which there was a sulfa drug that was being sold. And the sulfa drug, the manufacturers of the sulfa drug, in order to increase its palatability for children as an antibiotic, were incorporating ethylene glycol into the sulfa drug. And ethylene glycol is also known as antifreeze. Ethylene glycol, it made the drug sweet so that it was tastier for children to take this sulfa drug. Of course, if anyone knows about antifreeze and ethylene glycol, it is severely, severely toxic. And so after this sulfa drug was on the market, it killed more than 100 people. And that led to the enactment of this Federal Food, Drug, and Cosmetic Act, in which it basically set up the Food and Drug Administration and gave the FDA the authority to oversee the approval of drugs, devices, food supplements, food ingredients, and so on for the market. So that was a good thing. But unfortunately, at the time when this law was enacted, it required that all drugs must be tested on animals. Okay, so that was what the requirement was in 1938. And we could say, okay, that's all they knew in 1938. They only knew about how to test on animals. But the law was never updated since 1938. So the Food and Drug Modernization Act of 2022, of 2022 or 2.0, updates that 1938 law. And what it does is it simply goes in and changes the word animal in that initial law with non-clinical. This is something that I was doing when I was at the Food and Drug Administration for 10 years. And when I was there, for guidances, for documents, I actively would replace the word animal in any document or guidance that I was part of. I would replace the word animal, and the word I used to replace it with was preclinical. And the reason why I did it was because I wanted to help the FDA move away from this narrow focus of just looking at animal testing as the only kind of testing that can and should be done in the preclinical phase, or the testing that for which the decision as to whether or not to move a drug or a vaccine down the pipeline should be based. So this bill, the FDA Modernization Act, pretty much replicates what I was trying to do internally at the FDA. And so what it does is non-clinical testing now is defined as a test conducted in vitro, in silico, or in chemico, or in a non-human in vivo test, meaning animal test, that occurs before or after the clinical trial phase of the process. And it includes tools such as cell-based assays, microphysiological systems, bioprinted techniques, or artificial intelligence. Basically, it includes the use of human methods that are based, or methods that are based on human biology. So it doesn't take away animal testing, it doesn't remove animal testing, but it expands the options that conceivably a drug or vaccine developer could use when they're applying when they're working with the FDA to apply for their new drug or vaccine. Now, great news. As I mentioned, this bill passed incredibly fast, and it was amazing, right? And it makes sense that it should would pass, because it basically says, look, the world of science has moved beyond animal testing, or moved to include so many other innovative methods, and we need to catch up the language of the policies. We need to update the language of the policies to catch up to where the science is going. But the bad news about this bill, or this law, is that it really, in practicality, doesn't really change anything, because it still leaves the decision-making at the hands of the FDA. And so the FDA can always say that they've always said that a drug developer can apply using non-animal testing methods. But in reality, the FDA still prioritizes testing. And as long as they prioritize animal testing, quite frankly, that's what drug developers and vaccine developers are going to do, because their end goal is to get a product into the market. And if the FDA is going to keep asking for animal testing, then that's what the manufacturers will be doing. And so, so the FDA's policies have to evolve. Until it evolves, this process isn't going to really change. The other limitation of this bill is that you have to de-risk the process for drug companies. It'd be nice to think that there's going to be one drug company that's going to be courageous enough and be a pioneer and a leader enough to try to push a new drug or vaccine, and to try to get it approved without the use of animal testing whatsoever, and only based on the use of human relevant testing methods. I would like to think that there is a drug or vaccine company out there that will do this. So for all you pharmaceutical representatives, we're looking for that leader. But I don't know, quite frankly, of any drug or vaccine company that is willing to be that leader. So we need to find a way to de-risk the process, to make it something that's not so risky for the first vaccine or drug company to try to get a product to the market without animal testing. The other thing I wanted to point out is that the FDA, as I mentioned, does not prioritize animal testing. And let me give you an example. In their budget for 2023 from Congress, they requested $5 million for a new program called the New Alternative Methods Program. Now, that might sound like a nice, good thing. They're saying that they want a program on validating and implementing alternative methods, or what I call human relevant methodologies. But then that $5 million is only out of a total budget of $8.4 billion that the FDA had requested. $5 million out of 8.4 is nothing. So I see that $5 million request as just a way for the FDA to claim that they're doing something to help promote the use of better human relevant testing methods without really doing much in that regard. The good news, and I know my time is wrapping up, the good news about the bill. Okay, so even though the FDA Modernization Act in practicality doesn't really change anything right away, what it does do is that it has caused a tremendous boon in articles and publicity about where the state of science could go. And so these are some examples of some of the news articles that came out as a result of the passage of the FDA Modernization Act. The first one, the FDA no longer requires all drugs to be tested on animals. That was in NPR, Wired, Medscape, New York Times, and others have also been having more and more articles about the new state of science. And so what I think the FDA Modernization Act does do, what I think is a positive aspect, is that it's helping to change the narrative. It's helping to show a world in which I guess I would say it's messaging that there can be a world one day in which there is no animal testing. And so it's drastically cut a break and made a break from that old narrative in which we have to do animal testing to a new narrative. It's really pushing this new narrative that we actually could move into a world where there is no animal testing, where there are other methods being used, more effective, more scientifically and human relevant methods that are used instead. So I think that's one of the key benefits of the FDA modernization. It's actually starting to message that there are better methods, more advanced methods that are more human relevant, that can be used, and that it's starting to message that there can be a world one day. What that one day will be, I don't know, or when that one day will be, I don't know. But these articles and the publicity around the bill is starting to message that there is a world one day where we will not be using animals for testing. So my last slide is just a quick recap of some next steps. So we need to, in order to continue to make progress, we need to continue to obtain dedicated government funding for human relevant testing methods. And that may work with individual governmental agencies, or it may occur through policy changes, through the policymakers themselves. We need to work with pharmaceutical companies. They are our allies in this regard. Pharmaceutical companies, their end goal is to get a product to market as quickly, as cheaply as possible, and as effectively as possible. That failure rate in animal testing is not helping pharmaceutical companies as well. So we can view pharmaceutical allies and bring them in to try to understand how to de-risk the process, to incorporate more and more human relevant testing methods in place of animal testing, and to see where are the gaps, what other policy changes will help support their move away from using animals in experimentation. We need to change the granting process. Of course, the research is going to go where the funding goes. In the United States, for example, and this gets into academic research and not necessarily all drug development, but most of the funding towards medical research from the National Institutes of Health goes towards animal testing and not towards the development of more human relevant methods. So that needs to change. The research is going to go, if the granting changes towards more human relevant methods, the research will go in that area as well. We need to solicit feedback from scientists. What is it going to take for them to move into more human relevant testings? And lastly, we need to change educational programs to educate the next generation of scientists and create a large workforce that comes out from not having conducted animal experiments, but using more human relevant testing methods. And with that, I will end. Thank you so much. My apologies for going a few minutes over time.
Thank you so much, Aisha, for this brilliant presentation. Yeah, it seems all so easy when you explain it. The question is why isn't it going forward? Thank you so much for all this great ideas. Yeah, as I previously said, please place your questions for Aisha and all the others, other speakers in the chat. We come now to our second speaker, which is Professor Meryl Ritzkes-Heutinger. Meryl is Professor of Evidence-Based Transition to Non-Animal Technologies at the Veterinary Faculty of the University of Utrecht in the Netherlands. Meryl, thank you so much for joining us today, and we are looking forward to your presentation.
Thank you so much for the nice introduction, Tamara, and I will try and share my screen now. Can you see my slides?
Yes, it works.
Wow, fantastic. Thank you so much for giving me the opportunity to talk on accelerating the transition to an animal testing-free world and on the question how we can achieve this together. I've been appointed as a professor in evidence-based transition to animal-free innovations last year at Utrecht University. And so this is a totally new ball game, I think, within science, and I will guide you through the steps we are taking in the Netherlands and hopefully beyond as well. So I'm going to take you through the circle of my working life. How did I get here where I am today? Then some words on where the evidence is behind our actions, and we did a historical analysis of the legislative requirements. Then could we act more upon scientific evidence by, for example, using systematic reviews? And then last but not least, I will talk about accelerating the transition to animal-free innovations, which is a program in the Netherlands called TPI, Transitie Proefdiervrije Innovaties in Dutch, and Transition to Animal-Free Innovations in English. I will tell you a bit about what transition science is all about and whether we can expand that. So first of all, how did I get here, where I am today? On the top, you see my first literature review on atherosclerosis in the rat, which I did when in my last year at Vet College in Utrecht, and learning about quality in publications, which was poor, and also the translation of animal studies to humans was poor. And so I wanted to commit to better quality science and better animal welfare and better translation. And at the time, I still believed that animal studies translated well. So my PhD at Utrecht University was on refinement, so trying to improve quality of science and animal welfare. After my PhD, I moved to the Unilever company, where I was the head of the animal unit, learning about quality guidelines that industry uses. For example, the Good Laboratory Practice Guidelines, which was an eye-opener that industry had such high quality standards when doing studies. After 5 years in industry, I wanted to go back to academia, and I became the head of the biomedical laboratory at the University of Southern Denmark in Odense. You see the picture on the bottom of this slide. I also became professor with the aim to incorporate the 3 R's, replacement, reduction, and refinement, in the daily practice of the animal unit. After 10 years in Denmark, I moved back to the Netherlands and became the head of the animal unit at the Radboud University Medical Center, again combining it with a professorship, aiming to incorporate the 3 R's again in the daily practice. I founded the 3R Research Center in 2006 and found out after a number of years that striving for the 3 R's in itself is not an effective way of getting it implemented. So I then discovered systematic reviews and founded CIRCLE, which stands for Systematic Review Center for Laboratory Experimentation. And the results of systematic reviews showed me that the quality of publications of animal studies and the translation is quite low. And that made me decide to move fully to replacement. So I've come back now at Utrecht University and work with the TPI on accelerating animal-free innovations. One of the things I've come across, and Aisha also already talked about the FDA, where did this regulatory animal testing come from and what can we learn from it? So I teamed up with historians to look into that. And we saw that it's not scientific evidence, but health disasters force acute decisive regulatory action. The sulfanilamide elixir disaster, where people died, led to finally passing the Food, Drugs and Cosmetics Act in the US in 1938, after the act had been under discussion since the beginning of the 1900s. In Europe, we thought, well, that's the US. This doesn't happen in Europe. But then the thalidomide disaster happened in Europe, which led then to new animal testing demands for drugs for market authorization, which meant a rodent and testing in a non-rodent during pregnancy. The US escaped from the thalidomide disaster thanks to the FDA asking extra questions. So there was a time delay, and that's why thalidomide did not cause a disaster in the US. The questions of the FDA were not around the effects of thalidomide during pregnancy, so they were different questions, but still. Then where is the evidence behind all of this? There is a pharmaceutical company, Vanda, who filed a court case against the FDA because they had to do a chronic dog study. And Vanda refused to do this dog study because they said, we've already got a lot of human data, and these 10-month dog studies do not translate well to humans. So the court case went on regulations that are in place that do not really contribute to the safety of a drug. Unfortunately, this court case was lost, but Vanda never did the dog studies, but they get a special permission for prolongation of use for the drug each time a patient wants to do so. So I wondered, do we wait to act until there is a crisis instead of using good scientific evidence to act upon? But then what about the COVID-19 pandemic crisis actions, where vaccines were conditionally approved in 1 year instead of the normal 10? So what happened then? You can read this in the article in the journal Animals on the promises of speeding up. We have been looking at the Pfizer-BioNTech vaccine, what happened during the conditional market approval of this mRNA vaccine. Fewer animal studies were done, more alternatives were done, faster clinical trials were started, and because mRNA vaccines are so well characterized, batch releases can be done without animal testing. So there was a huge reduction for that case study, at least, in the animal studies, and it moved a lot faster. So I published a worldview in Nature where I make a plea to medical regulators to look beyond animal tests and also use flexible approaches as were possible during the COVID pandemic. So can we use scientific evidence a bit more, please? One of the methodologies is systematic reviews, which bring us the most complete and objective scientific evidence. You see the logo of the Cochrane collaboration, which is for clinical trials, which started in the beginning of the 1990s, doing routinely systematic reviews of all the evidence of available clinical medicine. Strangely enough, this methodology is not yet mainstream preclinically, in contrast to the clinic where everybody thinks it's normal to do this in evidence-based medicine. And we use so many animal studies to predict what is happening in humans and for human health that it is still puzzling me why we don't do this routinely preclinically also. One of the founders of the Cochrane collaboration, Sir Ian Chalmers, also founded the James Lind Library, providing literature to patient organizations. He invited Pandora Pound and me to make a historical account of what happened so far with preclinical systematic reviews in the UK and the Netherlands. What this article clearly shows is that systematic reviews identify the limitations of preclinical animal research. What was also very remarkable in the history is the parallels we see in the UK and the Netherlands. First, there is a lot of resistance towards new methodology, and now we see gradual acceptance of this methodology. We had a special funding program in the Netherlands by the Dutch health funding organization called ZON-MW, giving funding to Circle to promote preclinical systematic reviews. This funding program made workshops, coaching, and execution of preclinical systematic reviews possible. After the program ran from 2012 to 2020, ZON-MW wanted to know what the impact was of performing preclinical systematic reviews, and you can find this full article in +1 with Julia Manon as the first author. The impact of performing preclinical systematic review is quite impressive, changing the motivation of the person who is doing the systematic review to do better in future and to promote research quality for themselves, for their teams, and for their fields of science. One of the people I know from Cochrane, David Goff, told me once, after somebody has done a systematic review, he or she will do all future research better. After we published the impact study, then he said, your study has provided the evidence for this statement. We also did some interviews, or Julia Manon did those interviews, and I have selected 2 remarkable quotes from the interviews from people performing a systematic review. I was quite shocked how poorly designed some studies are while still being published in a high impact journal. Step by step, you can do a lot of research in humans that is also very helpful. I did a lot of animal experiments, and yeah, I'm not going to do that anymore. I'm not going back to animal experiments. So what results of systematic reviews have demonstrated is that the publication quality is low. The translation of animal studies to humans is also low, and this is all made transparent. This indicates there's a clear need for change. But we humans, we all understand that changes coincide with resistance. So it needs perseverance and managing transitions, and transition science is here to help us. What is transition science? You see here the multi-level perspective transition analysis that we use in the Netherlands. First, look at the figure on the right, where you see a lot of arrows and texts. Don't read all those texts. It is just to illustrate that all these processes influence one another. You see at the left bottom of the figure the word niche innovations. This refers to, for example, the development of organoids or in silico artificial intelligence innovations in the biomedical setting. In the middle of the figure, you see the socio-technical regime. That is how we do things in science, in policy, in industry, what our culture is like. Then on the top, you see the socio-technical landscape. That's the society at large. What does the society at large think about the use of animals in studies, for example, and what does politics think about that? What we try to do in transition analysis is to identify the barriers, the leverages, and the opportunities in the niche regime and landscape level. The goal is to identify opportunities where we can accelerate the transition. The Dutch Transition Program to Animal-Free Innovations, TPI, has a mission to come to better predictions without lab animals. The ambition is that the Netherlands, with the TPI community, can be a catalyst of the national and international transition towards animal-free innovations. This partner program was founded in 2018 and has 11 partners, and also recently Young TPI was established, so a next generation moving on to animal-free innovations. The TPI policy is there in conjunction with the 3Rs policy to ensure animal welfare as long as animals are still needed. Here you see the stakeholders that assemble on a regular basis. So the people that work with animal studies and with alternatives get together and discuss what we can do to make the transition move forward. So the universities organization, so academia is there, funders are there, NGOs like Proefdiervrij are there, the ministries are there, Young TPI is there, and so we try to get into prioritizing mutual actions. Of course, we can't do this alone in the Netherlands. So TPI is also looking for international ambition and activities and connect to international activities. My dog is agreeing with me, as you can hear. The European approach is crucial if we want to validate, accept, and implement non-animal methods, NAMs, which are also called new approach methods. So TPI wishes to start conversations with other EU member states about validation of NAMs, to find a common ground on what is needed to improve validation, and together to accelerate these national efforts and improve our chances to a European approach, and I would like to add a global approach. I will show this slide again at the end of my presentation, so you can scan the QR code then. 2 years ago, I was so happy to experiment with a multi-level perspective transition analysis with my colleague Ingrid Visseren from the Radboud University, where we organized a very experimental honors lab, so honors academy. We offered a course to students on accelerating the transition to animal testing-free medical science. The students had to apply with a motivation letter and a CV and a motivation talk. And then we worked together throughout the program and co-created the program. We have published this in the journal Animals with the title Identifying Key Factors for Accelerating the Transition to Animal Testing-Free Medical Science through co-creative interdisciplinary learning between students and teachers. It has been a wonderful experience with teaching like that and co-creating this program. And I've never seen students work so hard in my life. And the good thing also was that it resulted in writing a funding application together. In the Netherlands, we are lucky to have funding opportunities for these broad transition science projects. And 3 consortia were awarded funding for acceptance and implementation of animal-free models in safety assessment. So there is specific funding for transition science. The picture on the right bottom is the picture we took this morning of the core team of our SAFE project, Safety Assessment Through Animal-Free Evolution, where we're going to work on accelerating the transition to animal-free next generation risk assessment. That's a whole set of alternatives instead of animal testing, so a different way of thinking. And we're going to apply a transformative governance approach, which I will show you in the next slide. I'm now working at the department with Juliette Lechler, which you see on the picture on the right above. She is one of the persons responsible for the virtual human platform for safety, where a totally in silico model based on human data is going to be built with the aim to predict what will happen in humans. And she was also recently awarded a project called AVARA, which I won't go into now. So what is transformative governance? In transformative governance, the aim is to integrate transformations and transitions. We all know we have a climate crisis, an energy crisis, a biodiversity crisis. We just had a COVID pandemic crisis. All these transitions inform and influence one another. And what you do within the transformative governance research, you study the underlying causes and try to prioritize actions, do action learning, see where these actions lead to and learn from it and take the next steps. So we will do this the coming 4 years, which will be a very exciting journey, no doubt. So in our SAFE consortium, the background, aim and logic are mentioned here. We all know problems of animal testing have an ethical component with the animal suffering. There is also the technical component with the low predictability and the huge variability. We're going to address these problems. The aim is to contribute to the acceleration of the transition to animal-free safety assessment for chemicals and pharmaceuticals in the EU, including the Netherlands and the US, by enhancing our understanding of how to govern the acceleration. What are we going to focus on? We are going to focus on next generation risk assessment, since new systems for safety assessment are necessary instead of only one-on-one replacement of animal tests. And transition and transformative governance approaches are required to address the sectoral and societal barriers encountered by alternatives. So we need mixed methods research and flexible approaches to combine quantitative and qualitative research. This means we need interdisciplinary and transdisciplinary research. Transdisciplinary research is interdisciplinary research also reaching out to society, so connecting to society. What does society want and need? And we've seen that providing scientific evidence is clearly not sufficient to make real changes. This is an example I got from Thomas Hartung from the CAAT Center for Alternatives at Johns Hopkins. The monocyte activation test based on human blood was validated 25 years ago, now first gets incorporated in the European Pharmacopoeia. In the US, this is still not accepted. So why does this take so long? And that's why we need to connect to social sciences to study why and how we can accelerate. There are promising recent developments showing transition is happening. Sanofi Aventis at the Falasa conference in June 2022 announced their goal of global commitment to a 50% reduction in animal use from 2020 to 2030. We've seen the first clinical trial approved by the FDA without asking for new animal tests and based on NAMs alone. But this was, of course, an existing drug for a new goal, which is called repurposing of drug. So the drug was already tested, but is now going to be used for a new goal. But anyhow, no new animal tests were required. A recent article combining stakeholders describes how scientific evidence can be built on NAMs. And this is an article which describes the way forward. In Europe, we've had the European Citizens Initiative and the European Parliament asking for a roadmap for phasing out animal studies. The FDA Modernization Act was passed in 2022, and that's an important signal, especially as Aisha said, it happens so fast. So something is changing in the air, I think, so which is a good sign. The European Medicines Agency has changed their 3R working group to a working party, which means they have to accelerate in the field of replacement as well. And then ECHA, the European Chemicals Agency, is organizing a workshop on NAMs on the 31st of May and the 1st of June, where the possibility of the roadmap will be discussed. So progress is going on multiple levels. What the EU is also doing is stimulating education. I've been involved in 2 of these e-learning modules, which you can find on the Ed+ learning platform, the European training platform for laboratory animal science. And for example, the EU 52 module on searching for existing non-animal alternatives discusses that we do not only have the one-on-one replacement, so replacing one animal study with one alternative, but there is also complex replacement, combining in vitro and in silico data into new modeling. And then there's the possibility of rephrasing your questions, so avoiding animal studies altogether. And a very good example is included in this e-learning module. So I can really recommend you to take it, and it's for free. In the Netherlands, we've recently seen that the boards of the university organization have accepted a target image in moving towards animal-free education in academia in the Netherlands. So this ambition statement on innovation in higher education using fewer laboratory animals has just been published. And it is now the challenge to get this implemented. So using fewer animals in education and, for example, more virtual reality, but also making it happen that a different mindset will arise by showing that you can have education without animals. The other point that is important is legislation. I've recently been invited to get an implementation partnership with the University of Zurich, which is looking at the implementation of the 3Rs in the Swiss law in a project from 2022 to 2026. This project is dedicated to the 3R principle from a jurisprudential perspective. They will also look in comparative legal considerations, so compare Swiss law with European law, for example, and fortify conversation between stakeholders in science and practice. And one of the questions we already discussed is, what does it mean in practice that 3Rs are a legal obligation according to EU Directive 2010-63 EU? What does it really mean, and how do we explain this legal obligation? I sometimes wonder whether we need court cases for accelerating the transition. One of the court cases that was won by the Esso company was on the REACH legislation, where animal studies had to be performed according to REACH, where Esso said, but the chemicals legislation describes that animal experiments should only be done as a last resort. And they thought that was not the case here. And so they went to court and won it. So via that jurisprudential route, we maybe get better policy guidelines of what our legislation is all about. So all in all, let's make it happen together and internationally, globally, preferably. Phase out animal studies and embrace alternatives as soon as possible for the benefit of animals, but also for the benefit of humans. The science and technology are here to do so. So we need to walk new pathways. First of all, decision-making should become much more evidence-based. Then transdisciplinary research and education will help to connect all the stakeholders involved. The multi-level perspectives and transition science and transformative governance will help us towards achieving internationally harmonized legislation and policies. So I really hope we can work towards that together and accelerate what is already ongoing. And then thank you very much for your attention. I promise to put this slide back on. So if you're interested to connect to TPI in the Netherlands, please use the information from the ministry where an email address is given and a QR code to scan. So thank you very much for your attention, and I give the floor back to you, Tamara.
Thank you, Meryl. Thank you. Yeah, very impressive presentation. I really think it is very important that we have this kind of research, because it's, as you said, it's not only about the science anymore. The scientific evidence is quite clear. But we need strategies and really how to get to the transition. And this is very important to have experts like you. Thank you so much. So now I am passing the mic to our third speaker, which is MEP Tilly Metz from the Greens. And Tilly is not only a member of the European Parliament, but she is also chairwoman of the European Parliament's Intergroup for Animal Protection. Tilly, thank you so much for speaking here today, and the floor is yours.
Yeah, good afternoon, everybody, or shall I rather say good evening already? So I'm very happy to be among you today. I was asked indeed to give an overview of animal testing in the EU, even though thank you to Professor Dr. Meryl Wietskes-Holtinger. She did already mention some initiatives on the EU level. But I will really try to get also a point where we stand regarding the transition. And as a member of the European Parliament, and you said it already, I'm chairing the Intergroup of Animal Welfare, but I was also chairing and still chairing the working group on animal in science. So I have been following this subject indeed very closely. A short introduction, but I have to warn you now, after this very, very excellent scientist, I'm not a biologist or a doctor, I'm just a politician, so I will not give you now a 20-minute scientific presentation. So I did, however, study psychology, so we talked already a lot about the narrative today and also about the changing process, and this has been, studying psychology has also been quite useful in approaching this subject of the use of animals for scientific purposes.
Indeed, and it was already said, we face a lot of resistance and obstacles to change because many scientists, legislators, and regulators believe we still need to use animals for all kinds of scientific purposes. And I think this is like sticking to this tradition is probably the biggest obstacle to the phasing out. So now let's look where we are at the European level. Since 2010, we have, and this was already mentioned also before, we have a European directive with a declared end goal of total replacement of the use of animals in science. Unfortunately, Directive 2010/63 did not provide a clear strategy nor concrete measures to take for transitioning to innovation without the use of animals. There was no timeline, there were no milestones, there was no allocated budget, and not enough, and that is, I would even say, the biggest problem, not enough coordination foreseen. This might be one of the reasons why, despite the many scientific developments in non-animal approaches, the number of animals, and we saw it during the excellent presentation of Aisha, the numbers of animals used for science in the EU also are still extremely high, even 30 years later, 30 years after this directive. So more than 20 million animals are still used in the EU each year. 20%, you don't need to look at all those, just give an overview of all the figures. So, but I will tell you that 20% of all the scientific procedures are done for regulatory purposes. Fundamental research makes the bulk of the numbers, and 12% alone to cancer studies involving animals. And at the same time, and this is quite shocking, only about 0.1% of the Horizon 2020 budget, so the REACH budget, was dedicated to non-animal matters. For me, it is clear that any EU research investment and any scientific policy needs to be aligned with the 3 Rs. I think also most of us agree that a complete transition to non-animal science should be a way bigger priority than it is now. And the public opinion in the EU is also very clear on this issue, as very recent survey really also show. Whoop, I was too quick. So you see that 72% of respondents thought the EU should set targets and deadlines to phase out animal testing. 70% agree that replacing animal tests with non-animal methods should be an EU priority. 74% thought that animal tests for cosmetics and ingredients are unacceptable. And 66% even thought that the EU should immediately end all animal tests. And legislators and laboratories are currently way behind the public opinion. So the EU ban on cosmetic is a great example to show of how things can go wrong and fall really behind citizens' expectation because of a lack of awareness and consistency on the side of the legislators and regulators. Most people know that there is an EU ban on animal testing for ingredients in cosmetics, but not many consumers know that this is still happening in the EU and has been happening since the official ban that entered into force in 2013. As most of you probably know, the reason for this anomaly is an inconsistency with the EU legislation on chemicals. The tests on animals are not performed to meet the requirements of the cosmetic regulation, but for workers' safety under the legislation on chemicals. So the cosmetic industry is still required to test on animals in some cases, but the industry itself wants to leave animal testing behind for good. companies have adapted to the ban. They need clear rules, and once there are clear rules, they are quite creative, and they are committed also to the transition to alternatives to animal testing, because it is also what EU consumers want. I know that this test represents only a small percentage of all EU tests on animals, but to me it is so embarrassing and even shaming to disrespect a clear ban because of inconsistencies between laws and slow validation process and acceptance of alternatives. The ban needs to be respected. The EU most powerful direct democracy tool is, and it was also already mentioned, the European Citizens Initiative. So those are European petitions, if you want. And this has recently increased the pressure on this point. So we have the ECI safe cruelty-free cosmetics, and this collected in over 1.2 million validated signature. This is actually the second ECI on animal testing. The first one was Stop Vivisection. That has surpassed the 1 million signature mark. This shows that really EU citizens deeply care about this issue and that they consider current actions inadequate and insufficient. In September of 2021, the European Parliament took also a clear stance in support of citizens' call, and this was also already mentioned with a resolution. We adopted really an ambitious resolution on the phasing out of the use of animals in science, and you see the results there. 667 out of 705 MEPs voted in favor of the resolution. So a very clear majority, and I must say, honestly, I was positively surprised that it was such a big win. So a small working group of members of the European Parliament, which I had the honor to chair, worked for over a year to get this text adopted. We met with many stakeholders who provided scientific and regulatory input. We tried also to understand what concrete elements are necessary for an effective transition strategy. The resolution was not perfect, but still a strong call to action. Parliament made clear that the slow progress we have seen so far will not do for EU citizens. It also recognized the collective challenge of the mission at hand. In the resolution, the potential of NAMs is clearly recognized, be it in research, in regulatory testing, or in production. The slow validation and uptake of alternatives was deplored. We ask for more funding, training, and awareness raising of non-animal matters. We spoke already the issues of validation of the NAMs, but I think also, and somebody was questioning it in the chat, we never the animal testing had to prove so much their validation that the NAMs must do. So with this initiative, with this resolution, we hope to shake things up a little, to encourage member states, the Commission and relevant EU agencies to set up ambition and to increase cooperation. A big majority of members of the Parliament have recognized that we need a renewed and clear commitment to the ultimate phase out of the use of animals for scientific purposes, and very concrete steps on how to get there. So in the EU, a broad public and political majority has acknowledged need to move away from animal testing towards more human and human relevant models. But while there has been some movement in recent years, you mentioned the roadmap of ECHA. I could also mention the EMA guidelines when you use NAMs. And while some EU agencies, as I mentioned, them are encouraging the use of alternatives to some extent, we are still getting mixed messages from member states and the European Commission. I say mixed, even sometimes, as I showed before, quite paradoxical messages. Last year, for example, the Commission introduced questions in a public consultation survey on the revision of the REACH regulation, the main EU law on chemicals, implying like that new approach methodologies, so the NAMs, would weaken health and environment protection from chemical hazards and hamper the competitiveness of the EU industry. To achieve ambitious goals of the European Chemical Strategy for Sustainability, I believe exactly on the contrary, that the more dynamic approach is required in order to increase the accessibility and use of innovative non-animal testing strategies. A positive development which is worth mentioning, in 2022, the Commission updated the list of accepted testing methods under REACH to include new alternatives and to kick some animal-based methods off the list. So there is some progress. Of course, when it comes to regulatory testing, I always have to repeat, we all want safe products, effective medication, and high environmental protection. Though the goal is not to have less testing, research, or production, but what we want is different testing, new research, more human-relevant results, or equal levels of safety, but without, or even better levels of safety, but without the use of animals. But also in other non-regulatory domains, we know that we still have quite a lot to do for the acceptance and use of non-animal methods. What is clear is what we need is a better coordinated, cross-sectoral and EU-wide approach across all member states and all European agencies. Unfortunately, we are still waiting for a concrete action plan we have asked for in the resolution in September 2021. The European Commission reacted to the resolution but failed to present a concrete action plan. So the Commission response explained what is currently being done in the different fields for the transition to alternatives. It also explicitly welcomes the Parliament's aim to accelerate the transition to innovation without the use of animals in research, regulatory testing, and education. This is our common goal, the Commission says. Those are very nice words, yes, but I hope a concrete action plan with timeline, with milestones, and adequate budget will follow soon. Because we need a renewed and clear commitment to the ultimate phase out of the use of animals for a scientific purpose, and define steps on how to get that. If the US can do it, why not us? So what now? What are we hoping for? During the last meeting of the European Parliament's Intergroup on Animal Welfare, which I have the honor to chair, we discussed the ECI safe cruelty-free cosmetic. On this occasion, dear Luisa Bastos from the NGO Eurogroup for Animals summarized very well what needs to be happen now, what we want to see in an EU action plan for the phase out of animal testing. So her 4 points were: first, EU-wide collaboration and coordination. Second, clear milestones. Thirdly, identification of key research areas. And we need also very concretely new infrastructure to do the validation of the NAMs and to go ahead. Today, I call again on the European Commission to come forward with such an action plan, because it will be very difficult for individual member states to make great progress by themselves. We need a European plan and coordinated effort to leave animal testing behind us for good. To end my presentation on the 3 types of benefits that this would bring. So, of course, there are ethical benefits. The phasing out of animal testing is a progressive step toward a more compassionate, and I would say progressive society, improve public perception of science and technology. There are, of course, also economic benefits. Alternative methods are often, and it was already mentioned, faster and more cost-effective than animal experiments, make it an attractive option for the companies, and also making sure that certain medication, for example, comes quicker on the market. And lastly, scientific benefits. Alternatives can be more accurate and reliable than animal testing, leading to better drug development and human health outcomes. As I always say, we are not 80-kilo rats. I can't wait to hear the outcomes of today's discussion, and I look forward to meeting all of you again on future occasion. And of course, I will be staying with you still if there are questions or remarks. I tried to read a little bit in the chat because there was a very active discussion also going on there. But thank you very much for all of you for being there with us today on this very interesting and very important subject. And to be honest, one of my motivation to candidate again for the next European election is really the phasing out of animal testing. Thank you very much.
Thank you, Tilly, for the presentation. Yes, of course, it is not only important that we have researchers who work on the alternative methods, but also it is so important that we have politicians and decision makers, so people like you who can help us to achieve the goal of phasing out animal testing. Thank you so much. Yeah, so we are quite good at time, and we have already a lot of questions after these amazing presentations. So I'm now opening the question and answer session. And the first question would be for actually all 3 panelists, I would suggest. Even though they are incorporated in the legislation of many countries, it is difficult to implement the 3 R's principles in science and also to evaluate the effectiveness of their implementation. Do you have any suggestions for how to better do this, or have the 3 R's had their day? What does the ideal model of non-animal research governance look like? Any suggestions from both a legal and policy perspective are appreciated. Now we can discuss for half an hour about that.
I suppose I could start and say the 3 R's has definitely had its day. I would not at this point even focus on the especially the improving the welfare of animals. I mean, the truth is, no matter what, you're still experiments on animals are experiments on animals, and so the real focus should be on replacement of animal testing, because that's really what is going to change the entire biomedical field. Not focusing on the other 2, but focusing on replacement, putting the effort, putting the scientific effort, the funding, the policies into the discovery, the development, the improvement of human relevant methods that can replace animal testing, that will replace animal testing and be more effective. Because I think that we need to work on really that narrative, changing that narrative. We don't need more animal testing. So I would say especially the first R is irrelevant today. What we need is to replace animal testing. And so what we really need is efforts to help improve the methods that can replace animal testing and help discover the next breakthrough in medical science. Organ chip technology is one breakthrough that has come about, and there's a lot of great excitement about that. But that is just one. And of course, there could be other big breakthroughs that could come about. So not only do we need methods and policies that support the discovery of new methods, but also to continue to improve the methods that currently do exist.
Thank you, Aisha. Other comments on that?
I agree with Aisha. We should focus on replacement and new approach methods and new ways of thinking. And of course, the 3 R's are still in our legislation. So we need to explain what we need or what we mean by that. If there is an alternative, then our legislation says you're not allowed to do an animal study. And how are we going to check on that? And so complete literature searches and systematic reviews will lead to the implementation. So we also need steps in between on how to get there. And that will probably need discussions and explanations of what the law really tells us to do. What is a legal obligation if there is a replacement alternative? How have you searched the literature? Where did you look? How did you interpret that literature? That is not happening thoroughly at the moment. So that would be a step in between.
If I may add what I did not mention, and I think it's very important, it's by small steps and a lot of patience that we will have this phasing out. But what we are organizing now, I'm speaking on how to push on the legal side to have changement and a paradigm shift. What we are organizing now is a training for all the advisors that work for politicians like myself. I'm convinced of the NAMs, but a lot of other advisors working for the ENVI committee, the Environment, Public Health Committee, etc., don't necessarily are aware of all the NAMs that exist already. So we do a very basic training to explain about in silico, organ-read, working on cells, so all the possibilities that you have of non-animal testing that exist already, and that even, and it was mentioned by the scientists before, and even sticking to animal testing, and it was so good explained also from the side of Aisha, it blocks us to progress, and it is not efficient also often, as we said, to protect humans. So we do training for advisors, for politicians, and also of course members of the parliament can also join. And I would like to see legislation also a paradigm shift, which clearly would say it's a ban on animal testing if there are alternatives. That should be the rule for, and then to really achieve also a final total phasing out. I know it's not going to happen from one day to another, but if we would clearly say there is a ban on animal testing right now when there are alternatives, that would be clear signal also in direction what was mentioned, to have this narrative change. That was two things I was thinking about this question. Thank you.
Thank you. Yes, so a question to you, Tilly, also which fits here, I think. Why is the banning of animal experimentation for cosmetics still being relevant, although there was an official ban 10 years ago? Often asked.
Yeah, maybe I try to explain that, but maybe I know I sometimes go a little bit too quick, but there is a ban that is clearly there, a general ban on testing on animals for cosmetic ingredients that is there. But on the other hand, there was a demand coming from the European chemical agency, the ECHA, that was saying if the workers that manipulate the ingredients in order to protect them, there was so a paradoxical message, then you have to do animal testing. So it was an inconsistency between European rules. On the one side, the ban, a total ban on testing on animals of any ingredients that was used in cosmetics. And on the other hand, you have the rule, if there might be a toxicological risk for the workers, then you have to do animal testing. So there are the clashes of in order to protect the workers, and on the other hand, the general ban. And that's why I was very happy of the reaction then of safe cruelty-free cosmetic of the ECI, that really got a large support again from the citizens, that was really calling for, let's maintain the ban and let's kill this inconsistency that we have there. I mean, you could also ask the question, do we need to use such toxicology, such dangerous ingredients in cosmetics? And how many more new cosmetics do we need at the end of the day? Yeah? So it's really a bit asking about in what society we live in. But on the other hand, don't misunderstand me. I want the workers to be protected, okay, but with no testing on animals. We have alternatives. If there is still a need to test, and with the remarks I said before, if there is a risk that is so toxic, do not use those ingredients in cosmetic pasta. Voilà. And that's why there was this inconsistency, and that makes me very, very angry, because we had this clear ban and people are asking for that, and we should stick to this ban. And even the industry, as I said before, is asking for that. They say people, even it's an economic interest for them. They said we sell much better products where we can really label there is no animal testing being done on it. So even the industry did not want to go back on that, and they said we have the alternatives, we don't need this animal testing. So I hope I reply to that question.
Yes, absolutely. Thank you, Tilly, for clarifying, because this is really a question that we often get. People are thinking we have a ban, why is there a problem? Thank you for explaining. There is something that is also similar. This is, I think, to all of you. What about the outsourcing? I think it's great that EU, USA, etc. are moving away from animal testing, but a lot of companies just outsource their work to countries where animal testing is not banned yet. How can we tackle this, realistically speaking?
Well, I think one of the research topics is how these processes work within transition science. And so what are the leverages and the barriers and the opportunities to make moves? And if we can make the move to realize that new approach methods are better and more effective, more human relevant, then there is no need to outsource animal studies to other countries where it's cheaper, because then that would cost more time and effort, right? So I think that's one of the research questions that transition science will focus on.
Yeah, go ahead, Aisha, please.
No, okay, thank you. I was just going to say, in regards to the FDA, the outsourcing isn't quite relevant here, because if the FDA is asking for animal testing, the animal testing will be done. Doesn't matter whether it's here in the United States or elsewhere. If the FDA does not require animal testing, then, if you want to sell a product in the United States, and the United States is one of the, if you want to get something, the FDA only governs what products are sold in the United States. And I would say that almost every drug manufacturer wants to be approved by the FDA. And for better or for worse, FDA is sort of seen as a model regulatory agency, even though I don't think it should be seen as the model regulatory agency in many respects. There are some things FDA does well and many things they don't. But every drug manufacturer is going to want to get their product approved by the FDA. And so I don't think that the outsourcing issue is quite pertinent in this regard. I hope I explained that well. I don't know if I was more confusing or not.
Now, if I just want to add something, in general, during this pandemic, we realized that it is important, speaking about medicine and drugs now, that we need also to think seriously about relocalization of production of medicines and medication and so on and so forth. So indeed, the pharma industry clearly says to me, yeah, we prefer to produce outside the EU because a lot of standards are not so high in other countries, being on the impact on the environment and also on other standards. So first, I would say let's produce more medication and medicines again, also active elements in the European Union, so we have a better control on what's happening. And on the other hand, we should do here the same that we do in other sectors. We should implement like mirror clauses or ask for same standards, that we do not import, would it be medicines or cosmetics, where there is still, I dream of a future, where medicines that use still animal testing where alternatives exist. So that could be a solution in order to also push outside the EU for the phasing out of animal testing.
Thank you so much.
Of
course.
Hammer, if I could just add, just to clarify what I was trying to say. So outsourcing isn't a relevant issue with regards to FDA, because if you want to sell something in the United States, testing it elsewhere is not going to be sufficient. You have to still go through the process that the FDA requires. So it doesn't matter what other country has as far as their testing procedures or options, or what they allow and what they don't allow. You still have to have the FDA approve your drug or vaccine if you want to sell it in the US. So whatever FDA's guidelines are, that's what's going to apply for any product sold in the United States in regards to drugs and vaccines. Cosmetics are a different issue.
Yeah, thank you. Thank you for clarifying. Yeah, so I will now move to questions that are more about, general scientific things that people are also worried about. So I think this can be answered. Aisha, that was a question for you. I think you can answer it quite easily, because it's more, I think, a misunderstanding. Is it correct that you are advocating to move all substances untested to the clinical test phase? And do you think you will get enough test subjects for that?
Yeah, no, that is a completely misunderstanding. In fact, I would say that those humans who enroll in clinical trials are putting themselves in danger because of the non-predictive nature of animal testing that's been shown. Those facts and statistics I gave are facts that come from the NIH and the FDA and the Health and Human Services US government. Those are not my facts and statistics. That 90 to 95% failure rate are from our governmental health agencies. So the fact that animal testing is so non-predictive actually puts those first people who enroll in clinical trials with the current system based on animal testing at higher risk. But what I am suggesting is that to decrease that risk for patients and for people who enroll in clinical trials, let's use more human relevant methods in the preclinical, the before clinical trial stage. And these are, and I'm talking about complex physiological appropriate models. Organ on a chip technology is one type of model, but it's not just in isolation. You have a lung on a chip, a kidney on a chip, a brain on a chip. Believe it or not, even a mini brain. You connect them with a cardiovascular system, a neurological system, lymphatic system. You create a human body on a chip. We can go further then. One day there's going to be an Aisha on a chip, a me on a chip. using my cells and my tissues, so that you get to an era of precise, personalized medicine, where before a drug is even tried on me, you can see if a drug is going to be safe and effective in my body and for my disease. That is the future of science. That is inevitable. That is where we are going. And that is the type of testing method, one of the type of testing methods, in combination with other methods, that will most protect humans enrolled in clinical trials and all humans taking drugs and vaccines, and will most likely give us the answers we need to determine whether a drug is actually going to work and help us select the best candidates that can work and perhaps be cures for human diseases.
Yes, Roche, the big pharma company Roche, by the way, is working a lot on personalized medicine. This is an approach that they are focusing on a lot. So I think also this will be the future. Thank you. And another question, which is also similar, and what is your take, Aisha, that was also to you, and what is your take on fundamental research that helps understanding the human physiology and diseases? How do you manage metabolization requirements without animals? I think this is what you already explained.
Oh, sure. I was going to open it for if anyone else wants to jump in. Meryl, were you wanting to jump in?
Well, I think your previous answer perfectly explained that. You have organoids, organ on a chip, so you can combine organs from humans already. I've heard Donald Ingber from the Wyss Institute already connecting all these organ systems to one another, so you get a good prediction there. And also the in silico models with making calculations what will happen and predict. That I think will be the future.
Yeah. And so one thing we did not discuss, I would agree with what Meryl said, is one thing we did not discuss was basic research. I mean, most of our discussion has been on the research used for drug and vaccine development, for medical product development, not basic research, which is mostly conducted in academic centers and is a large part, if not larger part, of actually where animal experimentation occurs and how it, and under what category it occurs in the United States, at least. If we have a 90 to 95% failure rate with the applied research end, the drug and vaccine development, imagine what the failure rate is in the non-applied, where this is more curiosity-driven research with no end practical goal. And so what I would argue is that animal testing continues even at that stage, and especially at that stage, continues to hold back medical progress. If we want to truly understand human physiology, we want to understand how human diseases come about, we need to use models and methods that are based on human biology. It's not to say that we have never learned anything from animal experiments. Of course, we've for hundreds of years have been conducting experiments on animals. How could we not learn something, right? I can stare at a rock for 100 years and I will learn something. The question today is animal experiments the best way to get the answers we need? And the evidence, the overwhelming evidence is showing it is not the best way to get the answers we need, not today. And so there is a clear human health ethical reason in regards to human ethical reasons why we need to move away and embrace more advanced methodologies that are based on human biology. And if I can also add one thing, this is going to be a quick announcement. Center of Contemporary Sciences will soon be launching a large-scale initiative called Animal-Free Precision Medicine. And we invite anyone who wants to work with us, any organization, any representative from pharmaceutical company to please join us. We know we can get to that era where we have precise, personalized medicine, and we invite anyone to join us to help us get there as soon as possible. Thank you.
Can I just add that it's also important with human diseases to incorporate patients in the discussion. What is important to them? I mentioned the example in the EU Learning Module 52, that's on asthma, where asthma patients were asked, what is important to you? And then one of the top 10 that they gave was they wanted to learn to breathe better during asthma attacks. And so then all animal studies could be avoided because a clinical physiotherapy trial was started with a huge success. Some people even cured from their asthma. So it is also within fundamental science, what is important to patients when you're focusing on human diseases? Am I doing the right thing for patients? And so that can lead to avoidance of animal studies altogether, sometimes.
Yes. So one question that is also, that came a few times here in the chat, is I would like to know if there is any evidence, maybe a systematic review, to show that current norms have improved the 90 to 95% failure rate in clinical trials. Yeah. Who would like to comment on that?
I can't answer that. I don't think that's there already, but Aisha, do you?
How can we, I would say that until we actually get to a process where a drug or vaccine can be approved without animal testing, there's that evidence in totality is not going to exist. However, I would throw back and say, how can you be worse than a 90 to 95% failure rate? I don't know of any other scientific industry that accepts such a dismal failure rate. And I don't know of any other scientific field that does not advance from what it was doing 100 years ago. If you think about it today, no person would ever advocate for using the same rocket design that was used to get the first rockets up into space. Never would suggest that that's what we should be using today. Every other field advances. But for some reason with medical science, we cling to just this same old method and process that is more than 100 years ago. But on top of that, from my diatribe, away from that, there have been enough studies that have looked at pockets of failure rates in animal testing. And there was one that I would highly recommend everyone go and read. It was published by the company Emulate, which is one of the companies that creates organ-on-a-chip technology. And they basically looked at 27 drugs that had all been tested on animals, many of which had ended up having to be pulled from the market or received what we call a black box labeling because it caused severe liver toxicity, so much that it ended up killing many patients or causing the need for liver transplant. And so Emulate looked at their liver organ on a chip to see how well it could have predicted that liver toxicity in those drugs that caused a problem. And it found a sensitivity of 87%, meaning that all those drugs that passed animal tests that were found to be safe in the animal test, 87%, it had a sensitivity of 87%. It outperformed the animal tests in predicting which of those drugs were actually going to prove toxic for the human liver. So could you imagine if we had this method implemented instead of the very faulty animal testing? How many people's lives would have been saved? So that's one example. And there are many other examples out there that have shown more and more that these other methods are more predictive. Now, I will say one thing if I can. No one testing method is going to provide all the answers we need. No one testing method is perfect. But the fact that these are methods based on human biology already give them a leg up over animal testing. And it's going to take a combination of testing methods, organ-on-a-chip technology, imaging studies, clinical trials, microdosing studies, artificial intelligence, data mining, and so on, bioprinting using organoids, combination of different methodologies that's going to give us the best answer and the information that we need.
Yes. So I would have a last question, which is, purely the animal data that is the basis for this. So Dr. Akhtar says it's not the whole preclinical pipeline, but purely the animal data that is the basis for the decision to move a drug to clinical trials. That seems very implausible. Can she or someone else comment on that? And a similar question is, of the 250 compounds you spoke of, how many were actually tested in animals? For example, how many were discarded because of negative results in experiments with cell cultures, organoids, etc.?
I really want others to be able to answer as much as possible, and I don't want to take up the whole screen. But I did not say it was purely the animal tests that formed the decision. I said it is the basis of the decision, meaning it is predominantly the animal testing results that from which the decision is made whether or not to continue to allow human clinical trials. And that is, I say that from 10 years at the Food and Drug Administration and from my own experience working in institutional review boards that approved clinical trials or did not approve clinical trials. The institutional review boards primarily made the decision based on the animal testing results. So no, that's actually quite accurate. And then the other part of the question, I'm sorry, I forgot the other part of the question.
The second question, actually, that was similar.
Oh, it was about 250.
The 250 compounds, yeah, how many were discarded because of negative results in any like in vitro models, cell culture, organoids, and so on. So, yeah.
So I don't know what that 250, or if you're talking specifically with stroke or
I think
HIV.
it's in general, in general, why? I mean, is it only due to the animal experiments or also due to the failure of in vitro methods and organoids?
Oh, you're talking about the drug development process.
Yeah.
The slide you showed about the drug development process. I don't know. And this is proprietary information with drug companies, right? So they're not going to tell us exactly what drugs. This is a generic overview of the drug development process. I would like to know that information. If anyone is willing to share that information, I think that would definitely help with the discussion.
Yeah. Okay, so time is up, and yeah, we had, of course, a lot more questions that we could tackle here, and I apologize that we could not answer all of them, but at least we discussed, I think, some very important points. And with this, I would like to close the event. Thanks again to all the speakers, thanks to the organizers and also to our supporters, and to all of you, the audience, for joining, for listening, and for discussing and asking questions. And if you are in favor for a concrete strategy to phase out animal experiments in Germany, you are, of course, very welcome to sign our petition and also to disseminate it, because I think today we saw how many possibilities and opportunities we have to work on such a plan. So thank you all for being here, and I wish you all a good evening, and bye-bye.
Thank you.
Thank you.
Bye.
Bye-bye.
Thank you. Bye.